Hello, my name is Mauro Rezende and I work at the Institute of Tropical Medicine in Antwerp, Belgium as fellow researcher in computational biology. I'm here to present our work entitled Applying Metagenomics with Long-Read Sequence Data as Diagnostic Tool for Infectious Disease. A brief introduction on the main subject of the work. First, we know to identify the etiology of an infection, some specific laboratory tests can be used. Those specific tests are asked based on the suspicious pathogen and a set of symptoms. But there is a big overlapping about the clinical presentations of many infectious diseases. Therefore, several tests sometimes may be necessary to identify the disease agent, making the process expensive and slow. In addition, new pathogens can be missing employing that approach. Hence, metagenomic sequencing approach can be presented as a solution for this problem, once all the genetic material of a sample will be sequenced and analyzed. Nevertheless, the analysis of this data demands several tools and some knowledge on computational biology. Thus, we have been developed the Metatropics pipeline, which applies modern tools and long-read data from Oxford Nanopore as solution. The pipeline has as main targets its portability using Nextflow Work Manager and container systems such as Docker and Singularity. In addition, a graphical user interface has been developed using ShinyR library to make it accessible for non-bioinformatician people. On this slide, we can see the whole structure of the pipeline, which can be started using as input the raw data FAST5 from Oxford Nanopore sequencers such as MinION or the FASTQ files. Then a quality check is performed, followed by a filtration for human reads using MinMapTube. Then the reads are classified using MetaMaps tool and the virus sequence from HAFSAC database. If the classification returns a satisfactory result, a reference genome is selected and a consensus sequence is built from the mapping step for the classified reads using Medaka and IVAR tools. If the classification does not return a good result, the reads are assembled using Canoe and the contigs are annotated with BLAST and Diamond tools, along with NR databank from NCBI. Here we can see the command line interface of the pipeline, where it's possible to set different parameters which will change the behavior of the pipeline, such as the container system, use of GPU resource for basic calling and the multiplexing steps, minimum length, threshold for reads, and time window for Oxford Nanopore data. We can see now the graphical user interface being developed, where the same parameters from the command line interface can be set. Now I'd like to show an overview on the output files the pipeline is generating. First, the directory demult contains the FASTQ filtered by minimum read length. In the nanoplot folder, we have all the results from the quality control step using nanoplot tool. In addition, we have a multi-KC report compiling all the results for all the samples. The non-human FQ folder has the FASTQ files filtered for low complexity regions and human sequences. In the directory class, we have the results of the mapping step from MetaMaps against the virus sequence from HAFSEq. In the class AM director, the results for the read classification and sample compositions from MetaMaps. Class plot director stores the final results from the classification steps in a table with information coverage, depth, abundance, etc. And it has the plots for the sample composition and for each virus found in the sample. The consensus directory has the major consensus sequence for each virus found in the sample. And finally, the de novo folder has the file with the contigs and annotation reports for BLAST and Diamond tools. Here I'd like to show some examples of the results from the pipeline. First, we can see the multi-KC report with a quality check for the input data Here, the user can compare the samples with a reference to the number of reads, the quality of the data, and the read length. Here, a table with the main results from the classification step, such as the number of mapped reads, the fraction of mapped reads, abundance, the genome coverage, sequence depth, median for read identity, and mean for read length. We can see here in those plots some information on the distribution of the read length, read identity, and sequence depth for a specific virus genome found in a sample. And finally, we have a plot with the sample composition using Chrona tool to create an interactive plot. The pipeline is also producing an alignment between the consensus sequence and the reference selected in order to allow the user to check the quality of the consensus. The pipeline stores the mapping file in BUMP format, which was used to produce that consensus. Then the user can visualize the consensus quality using some alignment viewer for BUMP files such as IGV. As main conclusions, we have that Metatropics pipeline have been developed aiming performance and portability. It has already been tested for clinical samples producing valuable results. The graphical user interface will make it more accessible, and artificial intelligence will be applied to take automatic decisions on reference or de novo based approach to identify the pathogen. I'd like to thank all of you for your attention and all the Metatropics team.